Welcome back to the deep dive. Our mission today is really specific. We are going deep into the mechanics of why a mutation in one single protein,
Valosin-containing protein.
or VCP.
Exactly, VCP. And why mutations there don't just lead to neurodegeneration, but physically tear down the cell's most critical infrastructure, linking it directly to diseases like ALS and frontotemporal dementia.
This is a huge finding. I mean, for a long time, researchers have seen this common thread running through all these major neurodegenerative diseases.
You mean like ALS, FTD, even Alzheimer's and Huntington's?
All of them. And that common thread is a breakdown in something called nucleocytoplasmic transport.
Okay, let's unpack that right away. NCT is what, the cell's internal communication system?
You can think of it like that. It's the highly regulated shipping and receiving for the cell. It controls what goes in and out of the nucleus.
Where all the DNA is, the blueprints.
Right. And the cytoplasm is where the factories are, building everything. If that transport system fails, the cell is basically flying blind. And the physical structure controlling all this traffic, that's the nuclear pore complex, the MPC.
Exactly. The NPC is the gatekeeper. It's this massive, incredibly complex gateway that's built from about 30 different proteins.
And those proteins are called nucleoporins, NUPs.
Right, NUPs. The NPC is what maintains the barrier, but also lets the right things through. A healthy neuron absolutely depends on it working perfectly.
Which brings us to our main character today, VCP. You said it's an AAA plus ATPase chaperone. That sounds complicated.
It just means it's a molecular machine. It uses energy, that's the ATPase part, to grab onto big protein complexes and pull them apart.
So it's a disassembly tool.
A very powerful one. And when it goes wrong, the results are, well, they're devastating. These VCP mutations cause a whole spectrum of diseases called multisystem proteinopathy 1.
That includes FTD, a type of muscle disease.
Inclusion body myopathy, yeah. And Paget's disease of bone. But for our purposes, critically, it's linked to about 9% of familial ALS cases. One mutation, all these different problems.
Okay, so VCP takes things apart, and VCP mutations cause diseases where proteins often clump together. But here's the twist, and this is really the core of what we're looking at today.
That's the big reveal.
The healthy, normal VCT protein is actually responsible for quality control of the nuclear pores themselves. It's the maintenance crew.
Yes. And here's that aha moment for you right at the top.
Yeah.
Our instinct with the disease mutation is to think loss of function. The protein breaks, stops working, junk piles up.
But that's not what's happening.
Not at all. In VCP disease, the mutation causes a dominant gain of function. The enzyme becomes hyperactive, overzealous. It results in the excessive degradation of the nuclear pore. It doesn't fail, it gets too good at its job, and turns maintenance into demolition.
That is such a critical distinction. It's not about failing to clean, it's about actively destroying the house.
Exactly.
So let's really dig into that. VCP's dual role. Normal cleanup versus this pathological hyperactivation.
Okay, so let's start with the normal job.
The healthy maintenance crew. What's it doing?
Its day job is the selective removal of certain NUPs from the pore. Think of it like swapping out old or damaged parts of the gate to keep traffic flowing smoothly.
And this is especially important in cells that have to last a lifetime, like neurons.
Precisely. And we know this because the researchers basically broke the system on purpose to see what would happen.
In wild-type models, healthy ones.
Right. They used fruit flies, Drosophila, which are a fantastic model for this. They looked at their neurons and muscles, and they inhibited VCP's activity, either with a drug called CB5083 or just by reducing the amount of the protein.
And the whole maintenance system just stopped?
Ground to a halt.
Yeah.
When VCP, the extractor, was blocked, the NUPS parts like NUP98, NUP50, they just started piling up everywhere.
And not just piling up, right? They formed aggregates.
Ubiquitinated aggregates in the cytoplasm. It's a classic sign of a protein clearance traffic jam.
And this wasn't just happening in a petri dish. It had real-world consequences for the flies.
Oh, absolutely. This is why the model is so powerful. When VCP's normal function was blocked, the flies didn't just have messy cells. Their survival rate dropped. Their locomotor function, their ability to climb, was severely impaired.
So it directly mimics the motor neuron failure we see in diseases like ALS.
It proves that this routine maintenance job is absolutely essential for physical health.
What I find fascinating, too, is how specific it is. You said it was a selective process?
Highly selective. It's not a bulldozer. The data shows VCP primarily targets NUPs in what's called the nuclear basket and the central channel of the pore.
But not the main scaffolding.
Exactly. The core structural NUPs, like NUP93, were totally unaffected. VCP is a specialized tool, not a sledgehammer.
Okay, but in the disease state, it becomes the sledgehammer.
That's the perfect analogy.
So if blocking normal VCP makes NUPs pile up, what happens when you look at the actual disease-causing variants?
You see the absolute opposite. With the patient mutations like R152C or R155H, instead of accumulation, you see a significant reduction in the total amount of these key NUPs.
They're disappearing.
They're being destroyed. They saw this loss of NUP107, NUP98, and others across the board in aged flies, in motor neurons derived from patient stem cells, and even in muscle tissue taken directly from patients.
So that really cements the idea that the disease is caused by hyperextraction. It's a gain of function.
It has to be. The mutant protein is just pathologically overactive, and they found evidence for why. First, the mutant VCP protein is enriched right at the edge of the nucleus, right where the pores are.
It's hanging out at the scene of the crime.
It is. And it physically associates, it binds more tightly to NUPs like NUP98 compared to the normal protein. It's just drawn there.
And the fractionation data, that sealed it.
That was the smoking gun. It showed exactly what was happening. When normal VCP was blocked, NUP98 got stuck in the nuclear envelope. But with the mutant VCP, NUP98 was gone from the envelope and was instead found floating in the cytoplasm and nucleoplasm.
It had been ripped out and thrown away.
That is the molecular fingerprint of excessive extraction. No doubt about it.
Okay, so if mutant VCP is the engine of destruction, there has to be a guidance system. Something is telling it, go to the MPC, tear it down. What are the cofactors?
Right. VCP doesn't work alone. It uses these adapters or cofactors to find its targets. And the research pinpointed the UFD1-NPL4 complex as the key recruiter here.
UFD1-NPL4 is the delivery truck that brings the VCP wrecking ball to the job site.
It's the essential guide, and the proof here is really elegant. They showed that if you disrupt this delivery system, you can stop the destruction.
How did they do that?
Well, first they confirmed its normal role. If you reduce the levels of UFD1 or NPL4 in a healthy fly, NUP98 levels go up.
Because VCP can't find it to perform its normal routine clearance.
Exactly. But then came the crucial experiment. They did the same thing in the VCP mutant flies.
And did it stop the hyperdegradation?
It did. It was a spectacular rescue. Knocking down UFD1 or NPL4 in the mutant flies brought the NUP98 levels almost all the way back to normal, an 80% recovery.
Wow. So even with the hyperactive VCP mutation still there, if you cut off its guide, it can't do the damage.
You've neutralized it. And they went further. They showed that depleting UFD1 or NPL4 physically reduced the interaction, the binding, between mutant VCP and NUP98.
So you've got the whole chain of events.
The whole pathway is clear. The disease mutation makes VCP bind more tightly to UFD1-NPL4. That complex then drags VCP to the nuclear pore way too often, leading to this aggressive extraction and degradation of the NUPs.
Which brings us to the big so what question. Okay, the gate is falling apart. What does that actually do to the neuron that makes a person sick? What are the downstream consequences?
The immediate consequence is that transport fails. And the most critical piece of cargo that gets messed up in this disease is a protein called TDP43.
Right, and VCP disease is what's known as a TDP43 proteinopathy. That's the signature of ALS and FTD.
It is. Normally, TDP43 should be almost exclusively inside the nucleus doing its job.
But with the VCP mutants and the broken gates,
it's forced out. They saw a striking loss of TDP43 from the nucleus and a huge buildup of it in the cytoplasm. This was consistent everywhere they looked. Fly neurons, patient-derived motor neurons, human muscle. It's stuck outside where it becomes toxic.
And they drew a direct line from the NUP degradation to this TDP43 problem. It's not just a correlation.
It's a direct cause. This might be the most important link they made. They showed that if you just knock down NUP98 by itself, one of the key NUPs destroyed by mutant VCP, that alone was enough to make TDP43 mislocalize.
So a leaky gate is sufficient to cause the core pathology of the disease.
Yes, and it also triggered markers of DNA damage. The cell immediately goes into crisis mode.
And you can physically see the damage to the gate, right? It's not just theoretical.
Oh, you can see it. They measured the diameter of the MPCs in fly neurons and found they were smaller, they were structurally compromised. And in the human motor neurons, the barrier was leaky. Fluorescent molecules that should have been kept out were just flooding into the nucleus.
So when TDP43 is locked out of the nucleus, it can no longer do its job, which involves RNA.
Its main job is regulating mRNA splicing. It ensures the genetic instructions are read correctly. When it's gone from the nucleus, those instructions get scrambled.
You get what are called cryptic exons.
Basically, snippets of junk code get inserted into the message. And they could detect the garbage proteins made from this faulty code in both the patient-derived cells and the patient biopsies.
And this scrambling leads directly to the loss of two very specific, very important proteins for motor neurons.
Yes, STAthmin2, or STMN2, and UNC13. Both are absolutely critical for keeping axons healthy and letting synapses function,
So the chain is complete. Mutant VCP destroys NUPs,
which causes the gate to fail and TDP43 to leave the nucleus,
which causes splicing error,
which leads to the loss of TMN2 and UNC13 and ultimately motor neuron death.
which feels devastating, but it also points to a therapeutic strategy. If this is all caused by a gain of function, then inhibiting VCP should work.
And it does. That's the real hope here. Using that same inhibitor drug, CBA-FeOA3, it can rescue a lot of these problems.
What did they see?
They saw NUP levels get restored in the mutant motor neurons. They saw the aged mutant flies get their climbing ability back, a real functional improvement. And critically, it partially restored the levels of STM2 and UNC13.
But VCP is involved in so many things. Isn't inhibiting it across the whole body a bit risky?
That is the challenge. There's a narrow therapeutic window. You can't just shut it down completely. But what this study suggests is that you might not need to. You just need to dial back the hyperactivity of the mutant protein enough to stop the pathological destruction and restore normal maintenance.
And the fact that this rescue worked in flies, in human cells, in patient tissue.
That consistency is what's so powerful. It confirms we're looking at the true root cause of the pathology. The conservation of the mechanism is what gives us confidence that this could actually lead to effective treatments.
So as we wrap up here, the key takeaway for you, the listener, seems pretty clear. VCP is supposed to be a maintenance factor for the nuclear pore.
But these disease mutations turn it into a pathological demolition crew.
An overzealous enzyme that, through this UFD1-NPL4 cofactor system, just dismantles the nuclear gates, which directly causes TDP43 to go haywire and cripples motor neurons.
And because it's a gain-of-function toxicity, we have a clear therapeutic strategy. Inhibit it, bring it back in line.
You know, we started this whole dive by saying that these transport defects pop up in a whole range of neurodegenerative diseases, not just the ones directly caused by VCP mutations.
Right, you see NPC defects in models of Alzheimer's, Parkinson's, all over the place.
So that leaves us with a final provocative thought. Since the study so clearly maps out the exact mechanism by which VCP can destroy nuclear pores, could this pathway, this VCP UFD1 NPL4 degradation system, be a kind of final common pathway for damage?
That's the big question. Could it be that in other neurodegenerative diseases, cellular stress somehow activates this same destructive pathway, even without a VCP mutation? And if so, could carefully, precisely modulating VCP activity be beneficial for a much wider group of patients than we ever thought possible?
Keep asking questions, keep exploring, and definitely keep diving deep with us.